In this last part, I'm going to review a little bit about how the cell cycle is related to cell death. So if you remember, I talked about there are really two ways that cells can die. One is necrosis. So when we say a cell is necrotic, or tissue, usually we talk about our groups of cells as being necrotic tissue. Basically something happened that damaged the cell or starved that cell for the things it needs. And in necrosis, when we see it, what we know is that the cell is going to swell up, get really large, and basically just burst. So everything leaks out. And this triggers an immune response. So our immune system is going to respond, and with that, we're going to see some inflammation. So that's necrosis. That's something that's unexpected and related to some form of damage. However, in this case, when we see a different type of programmed cell death, in other words, this is a part of the scripting, really, of a cell, is if things are not correct, if the cell is getting old, if a cell just needs to be removed, such as when you're young and in development and you actually have some webbing between your fingers, and that is just part of the process, then that type of cell death is called apoptosis. And apoptosis looks different. So under a microscope, I would see when apoptosis is happening that the cell does something called blebbing, which basically gets a little bit of fragmentation and sort of then ultimately the DNA is broken up, the cell detaches from its neighbors, and then the parts that are then available are going to be absorbed by some of the healthy cells. So it's kind of a recycling. So that's apoptosis, programmed cell death. Now cancer is what we're about to talk about, and I'm going to go into this in a little bit of detail using something from Howard Hughes Medical Institute, which is to look at cell cycle and cancer. But when we think cancer, we think of a group of cells that are growing uncontrollably, very often related to some type of a gene mutation, and then that gene mutation or something else, and maybe in the environment, is going to cause a faulty protein, and that's going to not be able to regulate the cell cycle as it normally would. So we're going to take a look at that. Let's take a look at it. So here, remember that there are genes in the DNA of healthy cells. Some are called proto-oncogenes, which code for those things we say are positive cell regulators. Things which, when they receive the proper signals that everything's good, they're going to say go, and that cell is going to go through the process of reproduction. And there are tumor suppressor genes, which are also important. These are the stop signs that are genes in the DNA that code for cell cycle inhibitors, not allowing the cell to move into the next cycle until a lot of specific things are in order. So the one we've looked at already, one of them is P53, and P53 is coded by one of those tumor suppressor genes. If you remember, this is going to be an inhibitor normally. It's sort of putting the brakes on things, and it is regulated also by some of those cyclins that we talked about. So it can get disinhibited when everything is in good shape. But its job really is to be sure that there's no DNA damage. And so in the case where there might be a little bit of DNA damage, it's going to stop the cell from moving into the S phase, and it's going to, first of all, be sure that it's initiated some of the possible ways to repair the DNA, which DNA can do that. And then, if all is good, then it's going to go back and sort of cause the cell cycle to restart. But if it's not, it's going to stop the movement and instead lead to this apoptosis or programmed cell death. So that's normal. This is all the normal condition. Now, over here, you see what happens when it's mutated. So if P53 is mutated, it isn't able to read the signals, and therefore it doesn't stop the cell from dividing, which means it's just going to go right into the S phase, even when there's DNA damage, which can cause ultimately the thing we call cancer with uncontrolled growth. So something that is a tumor suppressor gene, such as RB, for example, and P53, these are the ones that are going to provide a break to the cell moving into the next phase. But if they have a mutation, and if, for example, RB has a mutation, then it may not be able to stop a cell from moving into the next phase. So essentially this is what we call a loss of function mutation. So normal cell division, we normally have a normal tumor suppressor cell, which is under the control of the cell cycle and will only allow the cell to move on when things are good, and it is coded for by this thing called a proto-oncogene. So this is the normal situation, and then we get normal cells. And most of the time that's true. But if we have a mutation in a tumor suppressor gene, or we have a mutation in a proto-oncogene, so this means that we can have either one of these, either the accelerator or the brake can have a mutation, and that can interfere then with what happens. We're going to get loss of function and gain of function mutations, which, don't worry about these, we'll talk about these later, which can lead to cancer. I want to go back here. Yeah, there we go. All right. So here we have how cancer cells are different. So basically, they ignore all of the stop signs and the checkpoints, and they keep going, they keep growing, and they grow abnormally. So here's a normal cell. Here's some cancer cells, which are large and can be variably shaped, and their nuclei, you can see, are quite large in there. They're much less organized. They just keep dividing and dividing, and they lose their normal features. They can't operate like normal cells. So again, checkpoints, remember, so we have a checkpoint here at G1. We have some other checkpoints along the way in the S phase and in the G2 phase. We're just focusing on a couple here. And then there's one here right in the middle during metaphase of mitosis that also is a checkpoint. And all of these things need to be paid attention to. And probably the most important one that we look at is this one here, the one that involves the RB protein, P53, and P21 restriction are negative regulators, and then also the CDK cyclin, which are those positive regulators. So abnormal cell development again. So if this is a healthy cell and we have normal, you're going to get healthy tissue. If it becomes mutated, you're going to get things that are abnormal and can lead to these tumors and sometimes be malignant, which is not good. Now there's a study that came out actually of a university in Nigeria that was really interesting, and this connects some of what we talked about on the metabolism, that cancer is, so this study by Fadaka et al. Basically kind of outlined what cancer is and how cancer cells change their metabolism in a way that's very interesting. And understanding how cancer cells metabolize glucose differently than normal cells is helpful, because sometimes that can be a way that we can target cancer cells to try to disrupt their ability to grow. And in brief, we know the normal cell respiration, a cell is going to take in glucose and it's going to go through all the different steps of cellular respiration. It's going to give off the waste products of carbon dioxide and water, but it's going to produce a lot of energy in especially in the last part of the cycle, that oxidative phosphorylation. But a cancer cell, interestingly, can do both, but it spends more of its time in that, more in the glycolysis, lactic acid. So almost when there's a lack of oxygen, it's going to act more like that anaerobic respiration. And that most of the time it's occupied making ATP this way, which means it's only going to be making a little ATP, but it's doing it a lot, like it's demanding a lot of glucose in order to get enough energy to keep growing and making more cells. So that's one thing that has been noted in cancer. And so here's another diagram. This is called the Warburg effect. And here's the glucose normally coming into a cell, and as you know, it goes down and creates a bunch of stuff, including pyruvate, and then normally pyruvate in a normal cell is going to go into the citric acid cycle. So it goes in the mitochondria, we get link reaction, we get Krebs, we get the oxidative phosphorylation. And then, in some cases, when needed, the cell can make lactate if oxygen is in short supply. But in cancer cells, notice that the arrow is leaning toward lactate. So it's going to make a lot more ATP this way. So a lot of the energy goes in here and much, much, much less into the TCA cycle, or the citric acid cycle. So just an interesting aside. So let me finish up now just talking a little bit about prokaryotic cell division. So prokaryotes are going to reproduce differently. They don't have, as you know, a nucleus. So basically they can copy all the information. They can take their DNA, make a copy, and then they basically split. And that's called binary, meaning into two. Binary fission, so fission is to split into two. And that's how they produce new individuals. Now they can do this in a really rapid fashion. So a single bacterium can become, millions within a day or two. So this is a very rapid form of reproduction. And so their reproduction of the cell is the reproduction of the entire organism, right? Because this organism is a single cell. Now your book does this, and it kind of walks through how binary fission occurs, and it brings up a particular protein. I'll talk a little bit about this in class, but just to show you how this occurs. Now I wanted to put in this, even though this is something that we might talk about a little bit more later, but one thing that's interesting is that here's the big double-stranded DNA that a bacterial cell normally has. But one of the things that a bacterial cell can do, here in this little loop here, remember this is called a plasmid. And so this is a little small loop of DNA. So here's a bacteria number one, and here's bacteria number two. And what bacteria number one is doing is it's creating something called an F pilus. And that is this little extension here that's going to connect with the bacteria over here that doesn't have that little plasmid or that little loop of DNA. And in doing that, it's going to make a copy of its little loop, and it's sending the loop over to this cell. So what it's giving this cell is some information, some genetic information that the bacterial cell number two didn't have. This is called conjugation. So it's a form of reproduction in which one cell can give some information to another. So let's say, for example, on this little loop, there was a gene to resist an antibiotic. Now this other bacteria now has that gene once this has been completed. So this is a way of transferring some genetic information from one individual to another. And in all species, from bacteria up through us, diversity makes you stronger. And so having this ability to give a new property to this this other bacterial cell that which then can go on and do binary fission is an advantage. So I think that's where we're going to leave it. So that's it. See you soon.